Hello, everyone. Thanks for tuning into my webinar titled What's in a Name? Taxonomic Updates to the Genus Lactobacillus and Implications for Probiotic Industry. My name is Andrzej Benkowski. I am the Senior Technical Leader for Probiotics and Dietary Supplements for Eurofins Microbiology Labs based out of Madison, Wisconsin, where we specialize in probiotic enumeration testing. So, outline of what we're going to discuss today. First, overview of the genus Lactobacillus, and then describe how the taxonomic update was made to the genus, what changes will be applied to both industry and academia, implications of the taxonomy change for the probiotic industry, and why the taxonomy change is a good thing both for the consumer and the probiotic industry as a whole. Okay, so let's level set and harken back to high school biology to talk about why any of this matters. Taxonomy is the branch of science concerned with the classification of organisms. Classification or categorization is a way we, as humans, make sense of our surroundings and is more of a function of the human mind than of the reality of life itself. For centuries, it has allowed us to understand the relationships between species. Invented in the 18th century by Swedish botanist Carl Linnaeus in his book Species Plantarum, binomial nomenclature is the system of nomenclature in which two terms are used to denote a species of living organism, the first indicating the genus, and the second a specific epithet. This way of naming organisms has been accepted by biologists worldwide and has stood the test of time as the way to name living organisms, whether they be animals, plants, or viruses. Classification is the ordering of items into groups based on similarities or differences, as seen by the hierarchical pyramid in the figure to the right. Taxonomy includes both nomenclature and classification. For bacteria specifically, since we cannot distinguish between macroscopic features like beak size or fur color, scientists have historically used physiological properties and more recently, genetic sequencing to classify and name these microorganisms. An additional layer of complexity in naming bacteria and viruses is the concept of horizontal gene transfer, when one gene of one species is absorbed into another organism's genome, which makes it difficult to determine how organisms are evolutionarily related. The tree of life, as we commonly knew it, subsequently turns into a web of life for bacteria. For probiotic bacteria, the health benefit is based on the strain, which is a subtype within the same species. Two different Lactobacillus acidophilus strains that are nearly genetically identical may purport different health benefits. Similarly, a particular strain of the species E. coli can cause infection, such as O157H7, while other strains do not, with many strains of E. coli even being beneficial and found in our microbiome, aiding in our digestion. So let's focus in on the Lactobacillus genus. The genus has been recognized since the 19th century, with it first being described in 1901 and demonstrating supposed health benefits in fermented foods as early as 1906 in a publication by Eli Metchnikoff titled The Prolongation of Life, Optimistic Studies, which implied lactic acid bacillus in Bulgarian yogurts as the agent responsible for preventing intestinal putrefaction and aging. The strain was later determined to not survive the gastrointestinal tract, substituting its lack of efficacy with the most extensively studied species of lactobacilli, Lactobacillus acidophilus. All previously grouped species of lactobacilli are gram-positive, rod-shaped, facultatively anaerobic or microaerophilic, non-spore forming, and acid tolerant. They were characterized by their signature biochemical trait, their ability to produce lactic acid as a byproduct of glucose metabolism. They are ubiquitous in fermented foods and beverages and commensal inhabitants of animal and human gastrointestinal tracts. Another key distinguisher between members of the Lactobacillus genus is whether they're homofermentative versus heterofermentative, where homofermentative organisms ferment sugars predominantly into lactic acid and do not produce gas, compared to heterofermentative species, which ferment sugar into lactic acid alongside other substances such as acetic acid and produce carbon dioxide gas as a byproduct. The genus Lactobacillus has been used as a way to preserve and ferment various foods and beverages for decades, but in reality, it's more like millennia. We just didn't know what was preserving the food back in that time pre-refrigeration. Species of Lactobacillus, among other lactic acid bacteria, are used for fermented dairy starter cultures, fermented vegetables, and fermented fish and sausage. What was originally a preservation technique, it has been shown these types of fermented foods may boost your health, and the term probiotic was coined. The good bugs, the beneficial microorganisms. The probiotic consensus definition was coined by the World Health Organization and the Food and Agricultural Organization of the UN in 2001, which is live microorganisms which, when administered in adequate amounts, confer a health benefit to the host. The keywords being live, the organism needs to be alive, adequate amounts, and health benefit, both of which are tied to documented evidence of a health benefit through the use of a clinical trial with a specific efficacious dose that is designated at the strain level. Remember, different strains of the same species can have completely different proven health benefits. So over the past few decades, as more and more species were being discovered due to the new tools scientists had at their disposal, the genus Lactobacillus ballooned from just over 50 species in 1995 to over 250 species by the middle of the last decade. The visually aesthetic phylogenetic tree on the right comes from the landmark paper by Jinshui Zhang et al. In 2020 that was used as the basis for the nomenclature change and reclassification. It represents a phylogenetic analysis based on the concatenated alignment of protein sequences for the 114 single copy core genes of the family Lactobacilliaceae. The different colors on the tree represent different phylogenetic groups with which the reclassification was derived. The color gradient in red represents the GC content of each genome sequence, with higher G plus C contents indicated by darker shading. The solid green circles represent the genome size, and the outer ring indicates the inferred natural habitats of the species as vertebrate host adapted in red, insect adapted in yellow, nomadic in green, environmental in blue, or unassigned in white. So with the categorization of all these new species of Lactobacillus, a problem arose. The genus became what is known as an umbrella genus. As species were being classified based on antiquated research tools scientists had at their disposal, they were being categorized as Lactobacillus. When more refined DNA analyses, such as whole genome sequencing, were applied to these same organisms, an extensive amount of diversity was discovered between species within the same genus. Some members of the Lactobacillus genus were more unrelated than others of different genera, including large differences in genome size and G plus C content. So now what? The genus no longer conforms to the nomenclature conventions we all know and love. New species of quote-unquote Lactobacillus were being discovered, and there was nowhere for them to go besides tossing them in with the rest of the Lactobacilli. A good analogy for the diversity is the species being as similar as a human and a marmot. There really was only one path forward. And that was to break up the genus Lactobacillus into 25 different and unique genera. The first suggestion for the reclassification came from a paper published in Probiotics and Antimicrobial Proteins by Elisa Salvetti et al. In 2012, titled The Genus Lactobacillus, a Taxonomic Update. In a follow-up publication in 2018, the same group presented a robust proposal to organize the genus Lactobacillus in 10 consistent subgroups using a number of different methodologies, including distance-based metrics, average nucleotide identity, average amino acid identity, percentage of conserved proteins, phylogenetic and network analysis on proteins, and phylogenetic markers. Zhang et al., whose phylogenetic tree I presented a few slides ago, had originally clustered the genus into 24 new subgroups based on single copy core genes involved in ecology and biochemistry properties, and followed up on this taxonomic suggestion in 2020 with their publication in the International Journal of Systematic and Evolutionary Microbiology titled A Taxonomic Note on the Genus Lactobacillus, Description of 23 Novel Genera, Amended Description of the Genus Lactobacillus Bierneck, and Union of Lactobacilliaceae and Leuconostocaceae. In the end, no true taxonomy exists, and depending on the methodology and cutoff used, different groups can be generated. In the end, the new classification was based on Zhang's paper with two key aspects: to create new genre with a better homogeneity that respects the defined rules for similarity between organisms in the same genus, and two, to create a stable number of genera that will accommodate future species without the need for further reclassification. So without further ado, here are the names of the 23 novel genera. The type genus Lactobacillus also remains, with its characteristic species being Lactobacillus delbrueckii subspecies bulgaricus, which happens to be one of two characteristic organisms of yogurt. As entertaining as it might be, I am not going to read them off to you for fear I wind up with a fox and socks moment, with my tongue getting twisted, mixing up the ticks and clocks with the chicks and the tox. So the new names can also tell us things about the characteristics of each genus. For example, Apilactobacillus is lactobacilli from bees. Limosolactobacillus are slimy lactobacilli, and some were named after some VIPs in the taxonomy world. The official document on the nomenclature reclassification can be a pretty entertaining read for the curious scholar. It gives information on origins of different species, many coming from fermented foods, which give insight into different regional traditions, such as fermented mustard and stinky tofu in Taiwan, kimchi in Korea, a wide variety of bacteria from Chinese fermented cabbage, kefirs, some sourdoughs, many different dairy products and fermented vegetables, and a number of drinks containing liquor lactobacillus, or lactobacilli derived from liquor or liquid, such as beer, wine, and sake. Reclassification of bacterial genera is nothing new. But in the past, it was mostly a matter of scientific discussion between taxonomists isolated in a meeting room, or a matter of safety concern linked to the identification of pathogens. Now, for the first time in history, the taxonomy change will have an unprecedented impact on the probiotic market, a category consumed by millions of people worldwide and projected to reach almost 70 billion US dollars of market value worldwide by 2023. You see in the graph to the left, the compound annual growth rate for the industry in the United States is forecast around 8% year over year. And there are similar growth trajectories worldwide as more and more research is published showing the wide array of benefits these microorganisms possess. With Lactobacilli being the hallmark genus of probiotic bacteria, I think something like over 80% of probiotics products contain a strain of Lactobacillus. They are widely administered in functional foods and dietary supplements to a very diverse population. The implications encompass the safety, efficacy, structure function claims, commercialization, and marketing of these products. And despite the unprecedented impact, the need for reclassification due to the continuous addition of novel species is required from the sheer diversity of the genus, as I described on previous slides. So in April 2020, the genus was officially reclassified. New genera that are recognized as containing prominent species in the probiotics market and commercially relevant strains were renamed starting with the same letter L to help mitigate some of the disruption caused by the reclassification on the industry. Keeping the name beginning with L and allowing abbreviations makes for a more streamlined, potentially less cumbersome transition. You see on the chart to the right some of the probiotic and starter culture species we are familiar with and their new names, including ones like Lactobacillus bacillus casei, Lactobacillus fermentum, Lactoplantibacillus plantarum, Lactocasei bacillus rhamnosus. And then there are some familiar names that have remained lactobacillus, such as Lactobacillus acidophilus and Lactobacillus helveticus. While this is obviously a big deal for the probiotic industry, the two key points that we need to remember and keep in mind if worries or concerns start to creep in on how the heck we are going to deal with this is one, the name change only involves the genus. The species and strain names remain the same. And two, arguably the most important takeaway is this is only a nomenclature change. It has no impact on the safety or efficacy of the microorganisms previously determined to be safe and suitable for use in foods and food supplements. Strains with generally recognized as safe or GRAS status from the FDA will retain their GRAS status. The clinical trial demonstrating a link between consuming a probiotic lactobacilli and gut health does not change. I added a link to a website put together by a collaboration of universities called Lactotax that is a helpful tool for finding out the new names. So the transition to the new names has already begun. We are over a year into the change, and you may have already noticed the new genre being used in, for example, research databases for comparing genetic sequences or on your ID test certificates of analysis from your identification testing laboratory. The QPS lists in the EU and GRAS status lists have already been updated. This flowchart is how I envision the transition, though in reality, there will be more moving parts and different levels and timings of adoption across the supply chain. For widespread adoption to happen, it has to start with the regulatory bodies worldwide. The probiotics industry is an international market with products and materials being imported and exported extensively. So harmonization is imperative to a smooth transition. Regulators work with industry associations such as the International Probiotics Association, or IPA, to establish timelines and grace periods for adoption. The strain manufacturers follow suit, updating the strain genus names, and from there, the rest of the industry, co-mans, finished format producers, brand labels, testing laboratories, and all of their corresponding documents are updated as needed. In conjunction with updates in the industry supply chains, education to the consumer is going to be imperative. Of course, the nomenclature change not only affects the probiotic industry, it has a very consequential effect on academia and the healthcare world. There are nearly 40,000 publications when searching PubMed that reference the genus Lactobacillus. New publications will need to account for both the historical and new genus names, while the residual use of the historical names is likely to occur in many books and previously published literature. Explanations may need to be given to editors and reviewers when citing the new name. In addition, literature searches will need to be conducted by searching for both the basionym or the old name and the new genus name. Likewise, lawyers and scientists involved in the validation of patent applications will have to deal with a nomenclature change for years to come, since the risk that previous patents could potentially be undermined by the use of the new name for the same claim. There are currently over 10,000 patents with the term Lactobacillus in the title or abstract, according to eSpaceNet. All new scientific publications and patent applications should be using the new name. While the new names should be used from now on, during the transition period, experts recommend using them in conjunction with the old name in technical publications. So something like the strain Lactobacillus rhamnosus GG should now be written as Lactobacillus rhamnosus GG, formerly classified as Lactobacillus rhamnosus GG. The bottom line, the name change can be manageable with time and dedicated resources, but comes with a significant cost to companies. So what all needs to change? Anything with the word Lactobacillus that now has a new name, including but not limited to informational materials, product labels, advertisements, import-export documents, regulatory dossiers, and approval and ingredient lists. As I mentioned earlier, previously published literature will not be getting updated. The dietary supplement category will be particularly challenging from a regulatory standpoint due to the lack of global harmonization. We are over a year into it, and trade groups such as the IPA, the European Food and Feed Cultures Association, and the International Food Additives Council are working with governments and industry to coordinate a global approach for updating all the documentation I mentioned. They are petitioning for a long transition period, think years, to allow the use of double naming in order to allow manufacturers and consumers the time they need to adjust to the name change. They are also requesting regulatory discretion on the use of the L abbreviation and are requesting that regulatory authorities update all lists that reference the former Lactobacillus species in a timely manner. They have also petitioned regulatory authorities requesting feedback for implementation and timing of the name change. So how will the change affect your brand? In summary, like I mentioned, the change as of April 2020 is globally applicable, but time is on our side. Many common species to the industry are not changing, and the species and strain names are not changing. The health claims associated with the strains are also not changing. Most commercially relevant species have new genus names starting with the letter L to allow for the use of an abbreviated name to help aid in the transition. Customer education is imperative to the success of the nomenclature change, and the fact that it is just that, a name change. It does not have affect anything except the label on the package. In the end, it is the same product containing the same organisms. And finally, industry associations such as the IPA are here to help your company with this transition. While it may seem daunting, the name change in the end will benefit both industry and consumers in a number of ways. The concept of shared mechanisms that may mediate health benefits is a potentially exciting outcome of the name change. The idea is that historically, structure function claims are defined to the strain level, but there is new research suggesting that these benefits may be derived from shared mechanisms that are incorporated among a specific number of strains, such as something like short chain fatty acid production. And these traits will be easier to distinguish with the new taxonomic groupings, as they're more likely to share these physiological traits. The new classification provides a better functional vision, which can improve the way industry develops new products. Downstream, there is an increase in discriminatory power linking particular properties to a specific genus, which can provide additional clarity to the consumer when shopping for probiotics for a specific benefit, such as gut health or immune support. Once the transition is complete, the new taxonomic situation will be more stable and will stop the unrestrained growth of the former genus. So to sum up, the Lactobacillus taxonomic update is only a nomenclature change, as I mentioned. It does not have an impact on the efficacy or safety of the probiotic organisms previously determined to be safe and suitable for use in functional foods and dietary supplements. It is a good thing. The International Probiotic Association, in partnership with the European Food Feed Council Additives and the International Food IFAC, is working with international regulatory bodies to inform and provide a rationale for the changes and request regulatory discretion for label enforcement, recognizing both old and new genus listings in the interim. While the implications for industry are wide-ranging, involving significant investment, the newly associated groupings will benefit future research and better inform consumers on the purported health claims. And we will need to learn from this transition and apply to the next one. Stay tuned because bifidobacteria are next. Thank you all very much for listening. I'll be out at Supply Side West. If you're there, stop by and say hello. We have a booth within the International Probiotic Association area, as well as a Eurofins booth. Come and say hi. Thank you very much for your attention and have a great day. Take care.